molecules on the target cell surface. These peptides are generated by proteasomal digestion within the cell, where HLA molecules are then loaded with these peptides and then presented on the cell surface. T cells come along and scan for particular peptides they may recognize, and upon successful binding, there's a signal generated downstream through the associated CD3 complex of proteins, and in particular the CD3 zeta chain. However, tumours do have an ability to downregulate HLA, and this is an example in cervical cancer where we can see in this particular tumour here there's a complete absence of one HLA allele. And this is thought to be part of an immune evasion technique where the tumours downregulate HLA so they become resistant to T cell activity and they can therefore grow. So the CAR approach basically short circuits this system. We take the CD3 zeta chain that is used for the predominant signaling of the T cell receptor and fuse an antibody fragment on the surface, and now this means that the T cell recognises cell surface proteins and avoids this HLA restriction. So the main advantage of this is that the T cell can now just recognise tumour antigens that are present on the surface of a T cell. The one downside is that we're now not recognising proteins that may be expressed within a cell. So CAR technology at the moment is primarily targeting cell surface proteins. It works. So this is just some work we published many years ago, just to prove a principle using in vitro cytotoxicity. So we take a gastric carcinoma cell line that expresses the antigen CEA and culture with T cells that are armed with an anti-CEA receptor. They're killed very nicely, whereas T cells armed with the same receptor, but then a single chain that recognises the NCAM, neurocell adhesion molecule, fail to recognise these cells. Whereas in the reverse, if we take neuroblastoma cells, which express NCAM but fail to express CEA, the NCAM-specific T cells now kill and the CEA-specific T cells don't. So this is just a proof of principle that killing is actually determined by the antibody moiety on the chimeric antigen receptor. Now, in terms of receptor, it's a fairly straightforward type 1 protein. There's a lump of protein that sits outside of the cell, which does the antigen binding activity. There's a transmembrane domain that anchors the receptor into the membrane, and then a signaling domain. So it's a fairly straightforward structure. I'm just going to talk about a few of these little bits that make up the receptor. The thing about the field developing is that people come out with all the various little funny things, so car mechanics, rules of the road, etc., taken from various reviews. So the targeting domain is primarily being derived from single-chain antibody fragments, as Sebastian mentioned just now, which is basically the heavy and light chains taken away from an antigen and fused, expressed with a short 16 to 20 amino acid linker. And so this single chain maintains the same specificity as that of the original antibody from which it's derived, the advantage being the small size and ease of expression of this particular single chain. However, we're also using single chains derived from phage display, and also actually other ligands work well as well. So, for example, receptor ligands or peptides fused on the end of a CAR also allow the T cell to recognise and target those particular targets. So we spent some time trying to understand whether there's some rules as to where the single chain has to sit in relation to the membrane. And we've come up with these fairly generic rules that for antigen targets or epitopes, such as here on the CEA protein, where the epitope is at the very amino terminus and at a fair distance away from the target cell membrane, as in CEA here and also on CD19, it appears that our chimeric receptor works optimally when there's no spacer region, so the single chain sits fairly close to the T cell membrane. However, if the epitope is buried away close to the target cell membrane, such as here in the NCAM protein or on 5T4 tumor antigen, then providing a spacer region to these particular CARs causes these to work better. So we hypothesise that there's probably an optimal spacing that's required to fit between the T cell and the target cell for optimal activity. But it's not an all or nothing effect. Basically, with or without a spacer, these T cells will work very well. So it's a very flexible system. Many different transmembrane domains have all been used, from CD8 alpha, CD4, CD28, FC gamma, and many others, and they all seem to be functional. In terms of the signaling domains, the first receptors that were used, CARs, just had a single receptor signaling domain, and this was primarily generated from the CD3 zeta chain. However, more recently, second-generation CARs have been developed where there's multiple signaling domains that are included. So the signaling domain from CD3 zeta, so the T cell activation signal, but now what we also include are co-stimulatory domains. So the T cells for full activation require two signals, signal 1 generated from the T cell receptor, signal 2 from co-stimulatory receptors. What we're doing in these particular receptors is that we're bringing both of those signals into the same protein. So one activation event results in full activation of the T cell. And indeed, a third generation receptor is, again, just an additional co-stimulatory domain. So, for instance, from OX40 or 41BB proteins. So 2 co-stimulatory domains and 1 activation domain. These all work. They all work fairly happily. So what I don't really understand is why they work so well. I haven't really found any particular receptor that doesn't seem to work in some way or another in a T cell engineered to express a CAR. I find that quite surprising considering the complexity of the whole T cell receptor activation process. But there we go. That's a good thing for us. So the process in terms of making these cells is actually much more straightforward now than it was, say, 15 or 20 years ago. We've been using predominantly retroviral gene transfer technology, and this is a fairly quick and effective method where we take peripheral blood mononuclear cells from the patient, generally in the form of a leukapheresis. They're activated into the T cells within that population are activated by the addition of mitogenic antibodies and some cytokine. Within 2 days, the population turns into around about 90% population of rapidly cycling T cells, where the retrovirus is then added in the presence of a substance called retronectin, which facilitates gene transfer. And after 1 or 2 rounds of transduction, the T cells are then expanded up for a period of 2 weeks. And so they generate a population of cells of which some express a particular chimeric receptor. And so for most of our clinical trial work, the threshold is 20%. So for a particular product going back to a patient, we have to achieve a transduction level of at least 20% for that patient sample to be able to be used. But there are other systems that are being used. So the retroviruses work efficiently, but lentiviral vectors now are coming on board, and the advantage of these is that the lentivirus can actually be added straight to the initial activating peripheral blood mononuclear cells. So in terms of a bioprocessing point of view, this addition of this technology is making basically life much easier to try and generate these complicated cell production systems. We're also now working with plasmids, RNA, transposons. So most of these technologies are all rapidly being developed to facilitate the transduction of primary cells and T cells, particularly in this case. So the general rules are that most CARs that are used, certainly in the clinic, are produced under an empirical design. Effectively, whichever the receptor is developed in a particular lab, whichever lab they like, they generally take that into the clinic. And there's still no consensus best receptor. Although the current view is probably that a second-generation receptor is the one that's most likely to be most effective in the clinic. Just about all formats result in redirected T-cell function, but in fact there's been very few head-to-head comparisons of different CARs. Varying gene transfer methods have been employed, so consequently there's no one single format that's been tested in the clinical environment. The important thing from the clinical environment in taking these cells is that we have to remember that the T cells themselves only represent one part of the entire therapy. And that's because the patients have to be preconditioned before they receive these cells. So this is based on the clinical paradigm that was really developed by Stephen Rosenberg at the surgery branch of the National Institute of Health in Bethesda. And they've come up with some general rules. So the patient has to be preconditioned. By precondition, this is generally chemotherapy, which eliminates white blood cells present within the patient. So this preconditioning results in removing of competing white cells, allowing space for our cells when we transfer them to move into. Also impacts upon the stronger suppressive factors that are present within the patient's tumour. So we're trying to give our T cells every opportunity to try and act. And it also causes some damage to the lymphoid compartment, which is recognised by bone marrow stem cells and stromal cells, resulting in the production of IL-7 and IL-15, which are homeostatic expansion cytokines, and so they can drive the expansion of T cells specifically within the patient. So preconditioning of the patient has at least 3 different impacts and really are important for this particular therapy. It's not clear whether giving T cells soon after preconditioning or a few days after preconditioning. It isn't clear whether, giving the T cell infusion soon after preconditioning is more important than giving it at a time point later. So 2 days later after preconditioning is less effective. But it really is unclear whether there's a cell dose. Some of the best responses have been seen with patients really receiving a sniff of cells, and this is because these cells will expand within the patient. It's also unclear whether post-infusion cytokine therapy is required. So for most studies at the moment, particularly the ones that we're doing in the UK, high-dose IL-2 is given routinely. However, it's highly toxic. Very few patients ever get through the whole course. And we're not entirely sure whether this is beneficial for the treatment. What does seem to be important is that readouts that correlate with clinical responses is finding these T cells resident within the peripheral blood. So if the T cells can be detected for a period of time within the peripheral blood, then better anti-tumor responses tend to be observed. and that's sort of logical. So a generalised outline in terms of how a CAR T-cell trial occurs. Basically, T-cells are collected from the patient, generally in the form of a leukapheresis. We take them into the cell production lab. They're activated, transduced with a virus encoding the chimeric receptor, expanded to large numbers, 10 to the 10 plus, and then given back to the patient who has been previously preconditioned. So to give you some idea of what certainly our early trials look like in terms of the impact upon the patient, in the early days we would do a test transduction first and then generate an eligibility report to confirm that the patient's T-cells could be transduced. We don't do this now because every patient transduces very well. We haven't had a patient that does not transduce with these particular systems. So around 2 weeks before the T-cells are given, the patient undergoes a leukapheresis. They're taken to the production lab where these cells are then activated, processed, and undergo expansion. They're transduced 12 and 11 days before treatment, and then 1 week before treatment, there's an assessment on the expansion and the transduction efficiency of these cells and some sort of functionality assay which satisfies the MHRA release criteria. And assuming that the T-cells are looking good, then the patients can then undergo their preconditioning. And for most preconditioning, this is a fairly toxic regime of cyclophosphamide and fludarabine. So this is more or less close to a transplantation sort of preconditioning regime. T-cells are expanded and then they're infused into the patient 1 day after the last dose of fludarabine. The patient then receives interleukin-2 and is monitored and receives prophylaxis to encourage stem cell engraftment, stem cell recovery. The reason for that is that after this treatment, so this is cyclophosphamide and fludarabine, this is lymphocyte count, but it would be the same for neutrophils. Patient lymphocyte counts become lymphopenic for a period of at least around about 8 to 10 days. So this means that they tend to be admitted for a median of around 16 days for a TIL, and generally range between 14 and 25 days. So this is an inpatient treatment, but it is an inpatient treatment because of the chemotherapy, not because of the T-cells. In terms of the number of the clinical trials that have been undergoing over time, so the first trial occurred around about 2006 or so, and they've been slowly making their way through. This has been published in Trends of Molecular Medicine, to the stage now that from 2010 to 2011, there's suddenly now greater than 20 chimeric receptor gene-modified T-cell trials all ongoing. And to highlight some of those, this is a list that we generated back in 2012, and this line here highlights targeting of CD19, so B cells. So targeting B cell leukemia and lymphoma is the favoured activity of people working with the chimeric receptor T cells, as you can see here by this many, and this is an underestimate of the number of trials that are currently ongoing in the world. So why is that? Why target B cells? Well, they've got a number of attractions as a target for a T-cell therapy. Firstly, B cells are located within the same compartment of the body as T cells, so it means that we don't have to get the T cells to travel into some weird and wonderful place to try and find the tumour. CD19 itself, the target antigen, is expressed purely on B cell lineages from pro-B cell to activated B cell, and it's found on the majority of B cell neoplasms. So if we target CD19 and eliminate all CD19 positive cells, we're not going to kill the patient. B cells also express ligands, co-stimulatory ligands, such as CD80 and 86 on their surface, which further enhances T-cell activity. And this B-cell depletion resulting from CAR T-cell activity is not life-threatening, and the patients can receive IV immunoglobulin to compensate for their lack of B-cells. So the upshot, so this was published from Stephen Rosenberg just last year, a review from Nature Review Clinical Oncology. And the important thing here is to highlight that the 3 trials that are carrying on at the Memorial Sloan Kettering in New York, National Cancer Institute in Bethesda, and University of Pennsylvania, regression of malignancy is reported in all of these trials that are ongoing at these centres. So this isn't a result of one particular centre. This is now being rolled out over a number of centres, and so it seems to be a really believable event. And this was going back to the report from Kite. We can see that they're achieving complete remission rates of around 61%, and partial remission rates of around 30% in large B-cell lymphoma. So that's really quite impressive. So, after targeting hemopoietic B-cell tumours, what about targeting solid tumours? Okay, so the first issue is that we need to worry about our CAR T-cells actually getting access into the tumour. They've now got to get out of the peripheral system circulation and be able to get to access tumours. The tumour antigens typically targeted by chimeric receptors. Remember, these are the ones that are on the cell surface, so we're now looking at generally tumour-associated antigens. And they may be expressed at high levels in tumours, but actually most of the ones that we're using are expressed at low levels on healthy tissue. So this starts to raise issues about potential toxicity. Solid tumour cells live in an immune-suppressed environment, and so there's generally a lack of expression of the sort of ligands that T cells like that may enhance CAR T cell function. And targeted T cell activity from CAR T cells could be life-threatening, and I'll discuss one example of this in a moment. And the immune-suppressive tumour environment really makes it a very unsavoury place for a T cell to be. So we need to think about how we're going to do this. And this is just comparing, actually, the difference between targeting a B-cell lymphoma and targeting a solid tumour in trials that we've carried out here in Manchester. So this is the first-generation receptor armed with an anti-CD19 chimeric receptor T cell, and this is measuring the number of T cells that are within the blood, and we can see that we can detect these out in this patient out for over 1 year. In a cohort of patients treated with exactly the same receptor, but all we've done is we've altered the CD19 single chain to one that recognises CEA, so we're targeting patients here with advanced gastric carcinoma. We can see that the T cells engraft for a very short period, but they're all gone within 14 days. So this is saying that we need to think about the sort of tumours we're targeting and how can we improve this system to go beyond this initial success with the CD19 CAR. Just to confirm with you that actually the patient who had that long-term engraftment did show a response in terms of reduction of tumour. So this is a tumour here, this lymph node has reduced quite significantly. So persistence of these T cells does seem to correlate with some poor clinical response. So in terms of targeting solid tumours, what sort of things are being done? Well, to aid tumour access, we're now engineering T cells with other factors, and things like including hormone receptors, such as chemokine receptors. So we can try and target these T cells based on specific chemokines that may be expressed from particular tumour sites. We're trying to identify T cells that have possess improved engraftment potential, or trying to improve the ex vivo methods we're using to try and grow these T cells. Now, bizarrely enough, we've spent many years trying to make T cells that are as active as possible outside of the body in vitro assay, but actually they're probably exactly the worst cells that go back into a patient, because what we want is these cells to go back into the patient, go through their normal lifestyle, and then go and develop an anti-tumour activity. So strangely enough now, we're working very hard to get T cells that have little activity in vitro, and then seeing whether they work in the patient. It's total reverse to what we've been trying to do for many years. The other issue is to try and optimise the patient preconditioning, which facilitates T cell access into these tumours. So at the moment, we're using a very old-fashioned, heavy-duty preconditioning, which has massive implications on the patients. Can we think of something that has less impact on the patients, doesn't put them into hospital for a week or so, but may actually impact upon the tumour? And these are the sort of things that are ongoing. In terms of targeting the tumour and the issues of trying to avoid healthy tissue targeting, we really need better targets. And there are some tumour-specific targets that are now being targeted, such as the EGFR variant 3, which seems to be expressed primarily on tumours and not expressed in normal tissues. And also trying to engineer the targeting of the single chain to only recognise cells expressing high levels of target antigen. Now, conversely, again, what this means is that we're trying to engineer the single chain to have a reduced affinity, so we can try and get down to a threshold where only cells that have high amounts of antigen on the surface will be recognised by these T cells. Again, it's all completely the reverse of what we've been doing for the last 20 years. There's another development is actually the fourth generation CAR, or we're now moving into trucks. So this is a cell that has a chimeric receptor. This is a second generation receptor. And upon activation, there's a signal that's driven to an NFAT, or a responsive promoter, that drives expression of cytokines located within the T cell. So this is a double construct containing receptor cell. The upshot is that we get activation of the CAR, which activates the T cell effector responses, but we're also starting to produce IL-12, which can enhance macrophage activity and differentiation. So these cells are being tested, and there's some activity that's being seen in the clinic that's been described by Stephen Rosenberg in Bethesda. So we've got encouraging beginnings, awaiting results from various trials, encouraging to date. Future developments will focus upon developing this therapy so we can give it an earlier stage to patients and how to avoid preconditioning. Solid tumour targets have been less successful to date. Achieving prolonged engraftment really is much more of an issue, and the target selection is really important. So we know that CD19 is probably the ideal target for a CAR T cell. The issue is that can we find something similar in solid tumours? I just want to mention about toxicity, because we have to be aware that these cells are very potent. They can cause major reduction in tumour volume, and they do that because they're very effective. However, we have to try and control that effectivity as well. And there's really four major routes of toxicity that's possible. One is that the CAR T cells undergo very rapid T cell activation that can drive a cytokine storm, so this can be potentially dangerous, and particularly a Th1-related storm, such as driven by interferon gamma, TNF alpha. The second mode is that this is an on-target, on-tumour, so where the tumour actually responds by being killed off and releasing lots of cytokines, and this can turn into a syndrome called tumoritis syndrome. On-target, off-tumour, so this is where the CAR T-cell may be targeting the antigen expressed on healthy tissue, and that can result in damage to the healthy tissue. And something that we've seen in models to date but not seen in the clinic is an off-target, off-tumour, where the CAR T-cells start a long process of interacting with other cells present within the mouse in this particular situation. And we've seen an increase in terms of myeloid-derived suppressor cells, resulting in a sort of granuloma formation. If we convert that, convert what we're seeing in the mouse to the patient, that may be something that may arise 2 or 3 years after CAR T-cell delivery. So it's something we need to be aware of, but it's only in the mouse model at the moment. Toxicity is an issue. So in the University of Penn trials, cytokine release syndrome is severe, particularly in this child who received CAR T-cells. And it turns out that IL-6 is the main cytokine that's causing the issue, and they managed that by giving monoclonal antibody therapy. There was one patient reported in Memorial Sloan Kettering who died as a result of actually sepsis, and so that was undoubtedly the preconditioning chemotherapy that caused the issue in terms of the treatment. But there is one report where a patient with colorectal cancer was treated with a third-generation receptor targeting the HER2 neoantigen, and this patient died of heart issues where the CAR T cells actually recognise very low levels of HER2 expression on the heart. So we need to be aware that on-target toxicity is potential. The issue here was that they went straight in with the most number of cells in a very gung-ho fashion. We need to be careful about how we organise our clinical trials to test these things. But there are ways to try and manage this, and these are all being tested at the moment. So one is a suicide gene system, so we include things such as TK, CD20, and caspase-inducing systems. And these are all being tested as a way of trying to eliminate T cells should you start to see some adverse events. Last thing then, to just talk about the practicalities of actually making these things. So up until 2010, we were making our cells for clinical trial at the National Blood Service in Manchester, just down by Plymouth Grove. This is a standard clean room where there's hoods and such all maintained within a HEPA-filtered environment. And during this period of time, so around 3 years or so, we generated a total of around 17 products that went back into the patients. We had 3 production failures. These were the first 3, of course, always the way. But after that, everything worked quite nicely. But the other issue is the fact that we actually need to try and be able to generate these things a bit quicker, because some of the patients progressed more rapidly than was considered, and so they didn't survive long enough to get their therapy back. So we need to think about how we can make these things better. And so that's led us on to developing and generating this new unit, so the Cellular Therapeutics unit, where we're now using glove box isolators that you'll be familiar with in industries to handle the cells, working with closed systems. And the upshot now is that in this unit, in one room using 2 of these isolators, plus bioreactor systems, we can get now probably up to the region of around 160 CAR T-cell products a year. So we looked at around 20 or so over a few years in the last system, and we're actually able to generate these things to a much more reliable manner. And this is just a wave bioreactor to show how these things are growing. So we're now moved, in the old days we were having to use 20 litres or so of media to get enough cells. We're now moving down to around 1 litre. It's sufficient to generate 10 to the 10 cells plus. So it works very efficiently. And those of you who work in clean rooms, you'll appreciate this is GMP aluminium foil. So the other thing that gets thrown at me a lot is actually the cost. And so these are ballpark figures. Don't shoot me if these aren't exactly right, but I know they're in the right ballpark for what we're doing at the moment. We think our cell processing costs around about £40,000 a patient, which, again, as an academic, sounds a lot, but when I'm told about how much monoclonal antibodies and such are, I find that to be quite competitive. And that's assuming a certain number of products going through the unit, and depends on gene transfer systems and the number of cells required. So I've got a number of caveats there, but we're talking about that sort of number. The clinical costs are probably around about £40,000 a patient, so it's the same as actually making the cells. So we think that the cost of this approach at the moment, bearing in mind it's a single infusion, is around £80,000 to £100,000, which I think is competitive in the current environment. But if we can improve our preconditioning, it means that these clinical costs would actually drop quite substantially. So the way forward really is to try and lose this preconditioning. So finally, clinical proof of principle is established through CD19-directed CAR T-cell therapy. So reports of durable responses in patients with advanced chemo-resistant leukemia. This has really kick-started the field that's been bumbling along for 20 years or so. But it's also come along at a time where we can actually generate these things now, generate these cells in a way that's actually more industrially relevant. To highlight that, methods of producing clinical grade T cells are improving rapidly. So isolator systems, we can grow 10 to the 10, 10 to the 11 CAR T cells for several patients at the same time in this particular room within a 7 to 14 day period. The important thing about the CAR is the fact that wide numbers of patients can be treated with individual constructs. So CD19 will target all B-cell leukemias and lymphomas that have the CD19 antigen on them. We're not restricted to HLA typing, for instance. And the modular structure of the CAR makes it very amenable to engineering. But we're still in the foothills here of trying to understand how CARs work and T-cell biology, so we can try and improve tumour targeting and reduce toxicity. And in particular, I keep harping on, but reducing the impact of patient preconditioning will reduce its impact upon the patient and, importantly, will significantly reduce the costs. So I apologise for going through a whole large area very rapidly. Robert Hawkins is the clinical PI that leads all of the work that carries on in Manchester, and everyone else here has all been involved at some stage or other in the clinical and the preclinical delivery of our T-cell therapies. And finally, just to acknowledge the various funders we have for all of this work. Thank you very much.